The views expressed in this podcast do not reflect those of our training institutions or the APA. The information discussed is intended for educational purposes only and should not be used for diagnosis or treatment. Enjoy.
Today on the podcast, we have Dr. Yasmin Escobedo Lozoya, an experienced researcher, teacher, and science communicator. She is currently conducting her postdoctoral fellowship research in Dr. Susan D. Mecky's laboratory in the Department of Genetics at Harvard Medical School, where she is investigating how specific serotonergic circuits and their plasticity may contribute regulation of defensive behaviors and coping strategies in mice. She holds a PhD in neuroscience from Brandeis University and has authored or co-authored 6 primary research articles. She was first trained as a researcher and communicator during her biomedical research studies at the National Autonomous University of Mexico, or UNAM. Additionally, she has mentored undergraduate researchers at Brandeis University and helped them conduct successful research thesis projects of their own. All right, so Dr. Escobedo, I always like to ask our guests how they kind of developed their interests. At what point did you know you wanted to study neuroscience in the brain?
I didn't specifically know that I wanted to do neuroscience in the brain until I started recording from single neurons in my last year of college in the lab that I was doing my last rotation and where I finally did my thesis. In that lab, I was tasked with what felt really daunting at the time. I was tasked with taking a spider venom and decomposing it into its components and then trying to figure out if any of those components had activity of blockade or of any other kind of activity on ion channels. So I would dissociate neurons and I would record from them, and then I would, add different purified components of the venom and then, measure the different ionic currents and try to find out whether they had, some activity. So that was really exciting for me at the time because I got to learn about every single ionic channel that was expressed in those cells, and I learned a lot about the in-depth biophysics of the channels, and it was just really fun for me to do that project. Yeah, so that's when I was like, okay, I really do want to do these, not just as an intellectual interest that I was always passionate about, but also like I just practically like to do these kinds of experiments. I like the kind of precision of what you can learn, right? And I like the fact that like the general interest of the collaboration was, let's try to see if there are some really great tools within these venoms that could be used to develop therapeutics that are very ion channel specific, right? And actually the toxin that I found was really interesting because it was a sodium blocker, and it was a voltage dependent sodium blocker, there was no characterized voltage dependent sodium blockers at the time. So I just really, like the idea of finding something that is a new kind of activity that could potentially be used for, like, there are so many disorders, including pain, right, that involve sodium channels, right? So the connection to the therapy just really got me hooked, let's say, onto professional neuroscience.
Awesome. So yeah, let's talk a little bit more about your research. So because we likely have listeners kind of from a diverse educational background, a lot of clinical trainees in psychiatry, could you begin by telling us about maybe the regions of the brain and the different serotonin receptors that you study?
So I generally study a system of the brain that is called the RAFE system, and that comprises the majority of neurons that produce serotonin, the neurotransmitter serotonin in the brain. And so that is composed of the dorsal RAFE, the medial RAFE, and the caudal RAFE nuclei within the brainstem. And they, well, as you know, or as you hinted at with the way that you asked the question, there are many serotonin receptors that are expressed in the human or in the mouse brain. I study mice. I don't specifically study serotonin receptors. I'm more interested in understanding the properties and the subtypes or the subdivisions of cell types within the RAFE system, right? And so they do express serotonin receptors themselves, They express a lot of a receptor called serotonin 1A receptor, which is been known or called the autoreceptor because it is expressed within the same cells that produce serotonin. They also express serotonin 1B receptors, which is a presynaptic receptor on the terminals of the cells that are synapsing onto serotonergic neurons of the whole system. And they express a little bit of the other receptors. And now within the rest of the brain, right, there's a highly specialized pattern of expression of serotonin receptors, with every single area having a very distinct pattern. I'm most familiar with the expression in the cortex because most of the processes that I'm interested in involve interactions between the cortex and the serotonergic system, right? And in the cortex, for example, there's a lot of expression of serotonin 2A receptors and serotonin 2B receptors, but it's really like, it's cell type specific within the cortex, and it's also region specific. Like the auditory cortex will have a different expression pattern than the frontal cortex or the somatosensory cortex. And again, and I'm talking about generally mouse here, but there's been recent studies by the Allen Lynn Institute in Seattle that have actually started to look at the distribution of serotonin receptors in humans and have given us much more granularity in terms of like which receptors are expressed and where and when within the human brain. So it's a very nuanced and complex field.
And I think maybe my question about serotonin receptors was a bit biased for my own experience of serotonin and thinking about medications that act on serotonin receptors. So it's useful to know there's this whole other side of the research that you're working on. So Dr. Escobedo, maybe it would be helpful for us to hear just kind of some of the technical aspects of your research. You know, what are you doing on a day-to-day basis? And I know that with the pandemic, maybe a day-to-day is more like week-to-week because you're doing like a week at the lab and a week at home. But I read that you're doing like intersectional genetics, or maybe you could tell us a little bit about some of those things.
So the way I like to explain the work that I do is that we're starting to really accrue some decent amount of knowledge about how different brain circuits talk to each other, right? For example, for learning and memory, you have heard about hippocampus, right? Which is a structure that is involved in essentially creating episodic memories or like tying together elements that form a context that can then be kind of like addressed, form an episodic memory in a person or in a mouse, presumably. So those are structures of the brain that are highly plastic, and to be able to form those memories, they have to communicate to many other places in the brain that are receiving sensory input, for example, that the structure is going to use to create relationships that form that context as something that you can store. And so the work that I do and the work that neuroscientists who focus on neuromodulation ultimately also do is to try to understand how neuromodulatory neurons are then essentially communicating with those brain regions that are involved in higher processes like learning or like transferring memories from a place to another, and how they can help structures that are creating those memories either coupled to each other so that they can pass information around, right? Or uncouple from each other so that they can stop the transfer or maybe even erase a memory. So neuromodulation is like a circuit that ends up acting more or less like a switch. So you can like really think of it as a form of the brain itself has built in to manipulate finely how its own circuits talk to each other. And so the whole idea behind a lot of the projects in the lab is to figure out what switches within the serotonin system go with which behaviors or functions, right? And how do we activate the right switches using our tools without activating pretty much everything else, right? And so a lot of the technology that is out there right now is just either turning switches on and off randomly that we don't even know exactly where. It's also turning them on and off differently in different patients, right? Like people with different population genetics in their background, right? That we don't really have access to as scientists or clinicians, right? Are going to respond differently to these drugs, right? And so that gives them different side effect profiles, and it's hard to manage. So what one could hopefully want to see in like the next 10, 15 years is like a development of both that population genetics background and precision medicine, right? And the development of better tools to target those switches more accurately.
Yeah, that makes sense. It made me think, I don't know if this has anything to do with what you were saying necessarily, but sometimes we do end up getting testing for, different patients, especially if they've failed multiple medications. We want to see, do they have differences in genetics and how they metabolize different medications and their different enzymes, their CYP enzymes in the liver, for example. Is that kind of something that's related to what you're talking about?
It's definitely like how you metabolize the drugs, or how do you metabolize the neurotransmitters themselves? Like what are the kinetics with which they get metabolized in the patient's brain could really affect the pharmacological intervention that you're trying to give the patient. Something about their metabolism that is specific to the patient, or at the very least to a population that is actually relevant to who the patient is, I think would be really, really key in having it through therapy. Just to touch a little bit psychedelics, because these are medications that are starting to become clinically available, right? And we know that, or we have what I would say is mostly just anecdotal evidence that they might not be useful in bipolar patients, right? But we don't really have evidence, and it is too dangerous to test it, right? So that also makes it so that we know very little about it, right? So if you make those medications more widely available, right, like one thing that you would want to know is if you knew something about not just the clinical history of the patients, but also their genetics, because like, let's say that there's bipolar disease in my family, and I also have some risk alleles, but I don't know about it because I've never been diagnosed with bipolar. Maybe my sensitivity to some of those drugs will be different than someone who doesn't have that background, right? And so maybe, if you actually have the genetic information coupled with treatment, you have to be able to start building on that relationship between the background of the patient and the effectiveness or lack of effectiveness of the treatment, right? And it may be, for example, that it's that there's a kind of bipolar disease patient that the drugs could potentially help, then only some psychedelics would work, and so on. It could get very, very complicated, but to really be able to have a more precise indication, you're going to have to need to use these tools in conjunction, right? Like the clinical practice, the genomics of the patients, and the advanced set of pharmacological tools to be able to cater a therapy to the patient.
Yeah. One other thing I was wondering about is, do you study BDNF, or I feel like that comes up a lot, like in our discussions about neuroscience as psychiatrists, brain-derived neurotrophic factor, and just how that's good for neurons and synaptic health. Like what's a more smart way of talking about it? How would you talk about BDNF?
So BDNF, as you say, is a really important neurotrophic factor for most neurons in the CNS. It can be released basally, but it also has, it's like a really complex gene that has many promoters that control its activity. One that is very well studied or fairly well studied because it controls the activity in response to calcium, which comes into the neurons when they're active, is promoter 4. And so we know that in many situations, if you have an experience that promotes learning, right, you will activate that promoter 4 and you would activate BDNF release in that neuron. And so there are many, it's a complex molecule because there are so many plasticity processes that BDNF is involved in, and there are so many like receptors that BDNF is involved in. And I don't specifically study BDNF, but it is known that serotonin neurons, for example, are very sensitive to BDNF release in places of the brain that they receive innervation for. And it is understood, how is the BDNF that they receive from the places that they're connected, the places that they're receiving information from, relate to either their activity or the properties of the neurons, especially in the adults, right? So I would say that it is an area of open investigation. And I'm sure that you're asking me about these because one of the, I think, perhaps favorite hypothesis about how depression works is that plasticity is being halted in the depressive state, right? Mm-hmm. And the application of SSRIs kind of like releases plasticity again through a serotonergic mechanism that increases the production of BDNF.
Right.
Right. So we don't necessarily know all of the steps that connect those molecules together. That's what I would say.
Got it.
But it is definitely quite involved in the biology of depression, or at least in the biology of some kinds of depression. And there's definitely a lot of support for the idea that at least some types of serotonergic neurons, especially the ones that are residing in the dorsal raphe, are involved in controlling the rate at which neurons, let's say, change or are plastic in response to environmental changes.
That's a really good explanation. Going a little deeper into, I think, like what I was trying to get at. Well, Dr. Escobedo, what are some other methods you may be using or have used in the past, that are maybe some big ones in neuroscience that would be helpful for us clinicians to know about?
I think that very helpful methods for you to start knowing about is there's a lot of new imaging technologies that have come on. And so for example, for my PhD and actually the research technician job that I was doing before, we were mostly using calcium imaging. And for example, the lab that I did my research technician job in, the Yasuda lab, was developing sensors for molecules other than calcium so that the molecule's activation could be traced like in real time while a biological process was happening, right? In that lab, we were interested specifically in the formation of memory and in a very specific process called long-term potentiation that we think it's involved in the creation of new memories. So in the Yasuda lab, we took a collaborator's construct and we modified it to be able to trace an enzyme called kinase 2. And that enzyme is one of the key enzymes that is required for the triggering, let's say, of the biochemical cascades that are required to create long-term potentiation in one single spine or synaptic connection in the brain. So the ability to be able to see that process unfold in real time and to know exactly when does it start and when does it stop and what are the requirements for it to be able to be triggered is super important to be able to understand how that process actually works. So in the Yasuda lab, he created, or basically like every postdoc or grad student would start to create an indicator or a molecule to trace the activation of different piece, right, of these components. So a lot of people were interested in a different kind of protein called small GTPase that are required for signaling how the cytoskeleton should move and change in response to different activity signals. So that kind of process of creating a molecule that can become fluorescent so that you can measure it in real time and so that you can do biochemistry, so that you can maybe follow the same process in an animal in vivo, but now applied to the signaling of neurotransmitter pathways, I think is a really important one to know about. So for example, at that time, I think that probably the Yasuda lab was the only one that I knew that was really, taking that on. So now there's a lot of other labs who have undertaken similar work and have developed amazing series of indicators of activities of different molecules. So for example, you can follow the activation of dopamine receptors, you can follow the activation of serotonin receptors. There's like a really, I think, new serotonin receptor activation tracer, right? So before now, to be able to know what the concentrations of serotonin were in the brain of an animal, you had to insert electrodes and you have to do a process called fast-scan cyclic voltammetry. This is a very technically challenging methodology, and also it's invasive. Or also you could do dialysis, which has a lower temporal resolution. But all of these techniques basically make it really hard to know what is the overall state, let's say, of release of serotonin in the whole brain. So with imaging technologies, it's becoming easier and more feasible to monitor the patterns of serotonin or dopamine or some other neurotransmitters release in a more global manner, and to better understand how concentrations of serotonin change in different behavioral states. So I think these are very, very powerful techniques. And then, of course, there's the calcium indicators have become better and better and better, and those are great ways to follow activity in the brain in general. So those are all, I think, things that would be important to be aware of and to understand what are the mechanisms by which they work. And that hopefully in the not so distant future will help us better develop therapies, right? That would better address the problems that different brains have.
Yeah, that's really helpful to know. I know it's kind of impossible for you to say, but how far away are we from some of these more specific, targeted types of therapies or treatments?
How far are we? Yeah, that would be very hard for me to say. Because, for example, like even if I told you that I have a plan to finish this research in 2 years, and that in 2 years I could tell you have to fix some problems on a mouse, right? Then the next step is to actually get it to work on a person or on patients, right? So that depends on clinical development, clinical trials, and it's a lot more complex because human beings are extremely diverse, right? So my mice are all as genetically identical as we can possibly make them, except for the genes that we engineer. But people are very diverse, right? And so, for example, that's another one of the kind of Pandora boxes that we have right now, that we, although there are very diverse patient populations, our clinical trials don't necessarily reflect those populations when they've tested for therapies, right? So that also makes it harder to predict whether therapies will be equally beneficial to everyone. So
right.
I'm gonna say I can't really answer that question right now. The other thing that we could say is that the effort to create coronavirus vaccines has created a lot of new avenues for scientific research to progress. More impetus, for example, for people to share their data through preprints, right? More impetus for the government and for the FDA to come up with ways to approve safe therapies faster. And then the third thing that I'm going to say is this is not related to coronavirus, but there's been a renaissance over the last 15 years of interest in drugs that have an effect on the serotonergic system. These are the psychedelic drugs. And there's been a destigmatization or a slow destigmatization of these drugs because of the successful clinical trials conducted over the last 15 years. And there are nonprofits that have been working for decriminalization and for greater approval. So although those drugs, those natural drugs by themselves may not be completely specific, they are opening a whole avenue of important pharmacology for the investigation, right? And I think that there's a lot of people who are very interested in them naturally because of how successful they have been with conditions that were considered quite intractable. So I would say that all of those things are probably going to accelerate the development of medicines and the interest in, for example, engineering the natural molecules into creating more specific compounds for, at the very least, for different receptors.
Yeah. And I'm really glad you made a point of saying that. And also the point about, sharing research and just how much coronavirus has shown us, the importance of working together and creating things that'll be helpful now rather than 10 years down the line.
Yeah. And so, for example, things like publication, like the first paper that I published in the US, it was when I was in the Yasuda lab. That paper went into Nature, and it took 2 years to actually, from the manuscript was submitted to when it was actually published. And, you could argue that some of that time was spent, semi-okay-ish, that there were things that the reviewers asked for that strengthened the paper. But you could also easily say it would have been better if the paper was published and people knew the results and they could have started to use those results to also inform their own science and to, advance faster. And that's more or less what's been happening with the coronavirus impetus for just publishing the preprints. So I think that to remove some of the kind of artificial, not even artificial, I would say almost like immoral problems of research by changing the way that we approach scholarship and research, it really is going to have a great and accelerating effect on the base at which we can do science.
Yeah. Hopefully that part is here to stay. Yeah.
Absolutely.
One thing I was just kind of going back to when you were describing side effects of medications like SSRIs, due to kind of like their non-specificity. One of the common ones I think about is nausea and vomiting because of the serotonin in the GI tract. Is that something you encounter at all in your mice, or that you study, with the serotonin in other systems beyond the nervous system?
That's a good point. We don't target the serotonergic neurons in the gut. The gut is actually pretty hard to study. It really definitely, it's a very fascinating place to study because of all of the interactions with, for example, the microbiome of the animals, right, or of people. In the case of people, it just has a immense number of different cell types with different neuropeptides. It is a fascinating place to study, but currently our lab is focused only on the serotonergic system within the RAFIS system, but that definitely would be a very important avenue to look at in terms of reducing side effects.
Yeah. And not to say you're not doing enough. Obviously, you guys are doing a ton of work just in the, relatively small system you're working within. So,
I think at this point, I feel like people have asked us like several times. I almost want to say that it's a question that almost always comes up. So I don't know, maybe someone should start looking at the gut serotonin system and see what we find that is interesting. There's definitely so much that we don't know, and it is fascinating that this is a neurotransmitter system or a molecule, right, that's been around throughout almost the entire tree of evolution. So it might be worth looking into.
Awesome. All right. Well, Dr. Escobedo, this has been really, really helpful. I think everything we can learn about neuroscience at this stage is just going to be more useful for us in our careers and working with patients. Is there any, last piece of advice or last words you want to share with us before we end for today?
Well, I would say I think that our generation of scientists and clinicians has a big and exciting role cut out ahead of us. I think that we're going to be the ones to hopefully finally be able to bring to fruition a lot of the work that our mentors have been building up and paving the way for us to be able to apply to the next generation of patients. So I would say that throughout the complexities of everything that we have to learn and that we have to apply, I just hope that we remain really engaged with the science and really willing to learn from each other as clinicians and scientists.
Well, Dr. Escobedo, thank you so much. This will definitely inform our clinician listeners and help us be able to understand neuroscience better ourselves and be able to explain these things to our colleagues, our patients, our medical students working with us. So thank you once again.
Thank you.